Well, hey AP Bio, hope you're doing well when you watch this video. It's always a joy coming to you live, or not really live, but alive from the internet. And so this is like the third time I've tried to make this video. I don't know what's happening, so we're going to try again. There's a lot of writing already, but I was like, I'm not erasing this all. But this is the Unit 5 guide from AP Classroom. Unit 5 is actually not very high on the percentage of the test, 8 to 11%. Unit 1, I think, is a similar percentage. So it's not the most crucial unit, but of course it's important and it ties into other units, of course, as everything in AP Bio does. It all is a big picture kind of umbrella. But 9 to 11 is just a general suggestion. And so you can always look over this stuff and read over. I encourage you to read your book, read these guides. We will be reviewing eventually. And we'll be going through these guides to do that. And so, but this is what we're going to be talking about in this video. I'll do a different pen color to see that. And so it is, just make sure it's still recording. Yes, and then we're going to go into genetics. So we're going to talk about meiosis first, then genetics. Chi-square is going to be a fun lab with M&Ms and stuff, but we got to understand how to like reject or fatal reject null hypotheses. We've been practicing writing null hypotheses this year and stuff like that. So it will be important. But yeah, meiosis is all about heritable information providing a continuity of life. Your DNA came from mom and dad, and then theirs, of course, came from their parents, and et cetera, so on and so forth, all the way, according to the Bible, to Adam and Eve. And so from one generation to the next, how does meiosis result in the transmission of this? Well, it's produced in haploid cells. And so you can see right here, I wrote n equals 23 and 2n equals 46. Well, this is our diploid number as human beings, 46, and this is our haploid number, 23. So sex cells, of course, would have 23, sperm and egg, 23 each, and 23 plus 23 is 46, rocket math here. And so that would equate to, Lord willing, human being, right? And so meiosis results in daughter cells, of course, half the number, haploid. Meiosis, too, the reason I wrote mitosis here, because it's very similar in how it works to mitosis. Meiosis versus mitosis, you got to be able to contrast them. Super important to be able to contrast them. And then Unit 2, again, similar enduring understanding. Heritable information provides for the continuity of life, but explain how the process of meiosis generates genetic diversity. How? And so homologous chromosomes separate. I wrote when right here. Like, when does that happen? We'll talk about it. You need to know that. You also need to know about crossing over. Like, how does crossing over work? How does it increase genetic diversity? And why is genetic diversity important? I mean, adding to resilience to the species. If the environment changes, if we have genetic diversity, it's crucial, right? To be able to have genetic diversity in case a disease comes or environmental changes, that we can, as a species, roll with those punches. Genetic diversity also allows us to fight genetic disorders and diseases possibly by having variation in the gene pool, right? Inbreeding depression. When, I don't know if you remember ninth grade, but the wolves on Isle Royale and the moose. There's 3 ways to do that: crossing over, random assortment, and fertilization. Okay, 3 ways to increase genetic diversity. And so let's go ahead and dive in. Is it still recording? My goodness, it is. Meiosis, yeah, creating sperm and egg. Okay, and so has anyone ever told you have like your mother's eyes or your father's hair? You look just like your brother or sister. You're like clones, right? And so here are here's a picture of my family. For time, I'm not going to dive into all of the things I usually say about this picture, but just notice a few things. First off, I have way more gray hair now. This is about 10 years ago. Thank you, teaching. And 3 beautiful young children. But also, my mom right here, this is her twin. And so they share the same DNA. But notice, they don't look the same, right? They look a little different. But they are identical twins, and when they were super young, it was crazy hard telling them apart. This is their mom right here, my grandmother. She passed away in July. My only grandparent passed away in 92. And so, Nana, and so, but notice, like in this picture, she has darker hair. And she always had dark hair, and that's a genetics thing. That's meiosis thing. But genetic variation in twins and siblings and cousins and, all of this is tied into this picture, okay? And so meiosis followed by fertilization ensures genetic diversity and provides variation, super important. That plays a role in natural selection. That's another reason why genetic variation is important, because natural selection acts on variation. Without variation, you have nothing to act on. And so, of course, using energy. So heredity is from one generation to the next, your parents to you. Variation, of course, is differences, and genetics is a scientific study of heredity and variation. So we're diving into genetics, probably one of the coolest topics in an AP Bio. I know it's not the most technically important as far as like a percentage on the test goes, but man alive, genetics is fascinating. Okay, so I would pause this right here, and we'll probably dive into some of this in class, but see if you can do these 4 points. Moving on. Genes are the units of heredity and are segments of DNA. Genes are passed on to the next generation via gametes. Gametes are sex cells, sperm and eggs. Okay. Most DNA is packaged into chromosomes. Those chromosomes contain DNA and protein. And so, for example, we have 46 somatic cells. Somatic just means like body cells. So your body cells, if they have DNA, not all fully functioning cells like red blood cells don't have DNA. They're just kind of like suitcases. The cells of the body except for gametes and their precursors. So each gene has a specific location or locus, not the car nor the insect, but locus on a certain chromosome. Okay, so asexual reproduction, of course, that is like you're creating clones genetically identical. This is not involving meiosis, right? It's produced from the same parent. So this is just like kind of separating without gametes. So that's not meiosis. Meiosis is sexual reproduction, which 2 parents give rise to the offspring that have unique, think about variation, combinations. And so here's some videos I'm not going to be able to show. Okay. So again, always do the checkpoint questions on your own. We'll be talking about them in class. Lord willing, I'll show you a couple of videos. And so the importance of meiosis, I've already said it probably like 10 times, but diversity. There are more than 70 trillion different genetic combinations possible from the mating of 2 individuals. Yeah, you read that right. Trillion. And males and females differ in the way they form gametes. Males and females differ in the way they form gametes. So in male, sperm production begins at puberty. But in the female, the process of producing eggs starts before birth and ends at menopause. For male, sperm production lasts throughout their entire life. Okay. And so meiosis, it's used for sexual reproduction. Parents, of course, are diploid cells, 2N, but we're making haploid cells. And so gametes fuse, nice little vocabulary word here, to form a diploid zygote. If events of meiosis go wrong, here's a little precursor to where we're going. Gametes would contain the wrong number of chromosomes, and that would be tragic, okay? Homologous pairs of chromosomes. In diploid body cells, chromosomes occur in pairs. We have 23 different types. Okay, and so chromosomes of the same type are called homologous. Prefix homo means the same, so they're the same length. They have their centromeres, which is like the central portion of it, in the same place. They show similar banding patterns. They have the similar genes, all of that. So same, right? Homologous chromosomes, the same. And so this leads us to the really cool science of making a karyotype. Okay, so this is made if there's like any sort of question about possibly chromosomal abnormality. This is also made sometimes if there was like problems with a developing baby or a miscarriage. My family, my wife and I, that was our first experience after finding out she was pregnant. Her first pregnancy ended in a miscarriage. Definitely a tough thing, and the Bible talks about stillborns and miscarriages, and it's been around for thousands and thousands of years, but it's usually due to like abnormal chromosome numbers. Okay, and so you can see right here we have 23 pairs. Here's a 23rd pair, X and Y, and so 1 2 3 4 5 10 15 22, and they're actually numbered based upon their size. Okay. And I know you're doing the meiosis disorder project, but there's only a few places, as you can realize, that the embryo can survive. Like if you have 3 2s, like if there's like another chromosome here, that embryo will not survive. Okay, and so that is homologous chromosomes. Okay, so you can see right there, this would be a female, and this would be a male. Y makes a guy. And so, and of course, we could dive into, I'm not going to in this video for time, but we could dive into the hot topic of like, sex, of as far as like, biological sex, male and female, and then gender. and is, they're a difference. And I just want to briefly mention right here, I mean, if you've come, to this video with questions or brokenness, Satan's first lie, and he's the father of lies, the Bible says, but Satan wants you to question God's design. Has God really said? Did God really mean to make me a boy? Did God really mean to make me a girl? Did God really mean, Eve, when he said, don't eat that tree? Like, isn't God holding out on you? Didn't God make a mistake? And so when Satan starts putting those lies into your head, it's a similar lies. And so identity lies and all of that, it falls under the same category of, man, like God made a mistake. But God didn't make a mistake when he made you. God created you and molded you together. And Psalm 139 is a great psalm about this. You are fearfully and wonderfully made. Wonderful are your works. And so you're a work of God, and you are wonderful, and you're created for a purpose. So that was free of charge. All right, here we go. So chromosome, let's just say this is dad or paternal, and this is maternal or vice versa. It doesn't matter. But you can see right there that when they duplicate, hence the duplication, there's just some terminology here. This is homologous pair, and then you have sister chromatids, which is like a duplicated chromosome. So when they duplicate, they turn into sister chromatids. If you can hear my son in the background, he just woke up from a nap. I'm going to have to speed this up. Okay, so sets of chromosomes in humans and human cells. X and Y, we already talked about that. The other two are called autosomes, 22 pairs, okay? So homologous chromosomes have genes controlling the same trait. The gene occurs in a duplicate, right? And so this is where, I don't know if you remember genetics, but this is where you get like an A, or like little a, right? But maybe dad was carrying the dominant and mom was carrying the recessive. That's what that means. And so there's two copies for each gene that you have, one from mom and one from dada. Yes, I just said that because I live in that world. Okay, variants, actually they're called SNPs, but that's, not for right now. The variants are called alleles as well. And so homozygous, could be like this or this. And heterozygous would be different, right? Hetero means different. And so there's a lot of different cases there. So let's say, oh yeah, they flipped it, blue for guy and red for girl. Okay, but maternal, right? So this organism has 6 chromosomes in its diploid state. Centromeres are in the central portion that's holding it together, proteins. Sister chromatids, so remember you have one chromosome and then it duplicates, and that's a sister chromatid now, and they're joined together, the centromere, okay? And then they'll join up in meiosis, okay? And so we'll talk about that here in a second. So that's just describing chromosomes. Sister chromatids, 2 non-sister chromatids, right? Non-sister, non-sister, and then they'll join together, forming a tetrad here in a second. And so do the checkpoint question. I'm not going to read it. So meiosis is a reduction division, which means it is trying to half the number of chromosomes. It involves 2 nuclear divisions. Meiosis 1 is the way more complicated part of it. And so homologous chromosomes pair up in synapsis. Chromosomes may recombine or exchange genetic material. Oh man, increasing variation, right? The 2 members of homologous pair separate. We'll look at that picture in a second. And each daughter cell receives 1 duplicated chromosome from each pair. And this right here is when we have reduction, going from diploid to haploid. And so meiosis 2, similar to meiosis in some ways. DNA is not replicated. Sister chromatids separate. 4 daughter cells contain 1 daughter chromosome. Each daughter chromosome consists of a single chromatid, and the daughter cells are haploid. And that's our goal, right? So let's look at these pictures. Again, pictures are the best way to kind of understand this, and we'll be molding Play-Doh to create like a stop motion video to help us understand this as well. But here we go. So pair of homologous chromosomes in a diploid parent cell. You can see after duplication, so this is like interphase, right? Cell cycle. Well, let's review. Ooh, man. Most of the time, the cell cycle is in interphase. And then, that consists of G1, S, and G2. Growth, DNA replication, checkpoints, all of that. And then you have mitosis. And then that would be cytokinesis when 1 cell turns into 2. I know that's horrible, but cytokinesis. Okay. Mitosis involves prophase. metaphase, anaphase, telophase. Okay? And so what happens in meiosis is you have these guys over time. You notice how there's just a blue or a red. That's already haploid cells. So this cell is going to have just dad's DNA. And this cell is just going to have mom's DNA for chromosome 1 or whatever. And then notice that they separate, the chromatids separate meiosis 2. Okay? So prophase 1, and then we're just going to go through the phase of mitosis. Okay? So it starts with interphase. Prophase 1 is one of the most important. I mean, they're all important. But prophase 1, of course, the nucleolus is disappearing. Each chromosome is duplicated already in interphase. Homologous chromosomes pair up. So notice, you see how they're pairing up. And they're called a tetrad or bivalent. Good important vocabulary there, okay? And right here is where crossing over happens at chiasmata. I think I said that right. So these are portions where the DNA is swapped, increasing variation, okay? And so here's metaphase 1 lining up in the middle. So homologous pairs, look at, so you see how they're lined up, the tetrads are lined up in the middle, okay? On the metaphase plate. And then anaphase 1, very important that you underline this, but anaphase 1, homologous chromosomes separate, not sisters. See, in mitosis, the sisters separate there. The sister chromatids remain attached, very important. So you can see right there, the homologous chromosomes are separating, but notice, you see, red and blue, blue and red. That is due to crossing over. So we have genetic combinations, gene combinations that were not present in mom or dad, now present in this cell, which gave rise to possibly, you or your sister or brother. And so homologous moves towards sister chromosomes do not separate. Until phase 1, daughter cells have one duplicated chromosome, and then it separates, and cytokinesis, it splits. Okay? Interkinesis is between, similar to mitotic interphase, except it's usually shorter. DNA replication does not occur, right? And then stages of meiosis, this is just the second part, meiosis 2. Very similar to mitosis. The chromosomes condense. The chromosomes align at the metaphase plate. The sisters separate, that's important. Always questions about anaphase 1 and phase 2. What separates in each one? Again, 1 is homologous chromosomes, and 2 is when the sisters separate. Are we still recording? Because if we're not, I'm literally, oh, sweet, we are. Okay, and then telophase 2 and cytokinesis, 4 haploid cells. So here we go. This is meiosis 2. Notice that they separate in the middle, and then they cross over. I'm sorry, not crossover. Separate to the poles. Okay. And so here's the stages. Here's just another picture. And we'll be practicing this in class, watching some videos about it, nonetheless. And so here's the comparison. And this is probably where we'll, compare. Let's just see where we're going here. Yeah, we're going to stop before part 2 for this first video. Okay, so mitosis and meiosis, the comparison. Mitosis. First off, the goal is clones. We're trying to create clone cells that look exactly the same. So metaphase, notice that individual chromosomes line up. In metaphase, pairs of homologous chromosomes line up. Also, crossing over happens here in meiosis. Crossing over does not happen in mitosis. Okay, so you have tetrads forming in meiosis. See the 4? No tetrads forming in mitosis. And then sisters separate, and that's good. In anaphase 1, the homologous chromosomes separate. Crazy important that you know that. Homologous chromosomes separate in meiosis 1 during anaphase 1. The sisters remain attached at the centromere. And then sister chromatids separate during anaphase 2. Crazy important that you know that, okay? Again, very different looks, right? Clone cells here. 4 cells. Well, let's just keep it in just the 1, right? Since we just did 1 division. We have 2 cells, but notice we only have homologous chromosomes. And there's only 3 chromosomes in each one. Now, you're tempted to possibly think, here's a misconception. Glad I caught this. You're tempted to think, no, there's not. There's 6 chromosomes. No, that's not correct. When they're joined together, they are one chromosome with two sister chromatids. That is just biology being biology, okay? So don't be tempted to do that. Haplo number's 3, okay? Summary, if you can walk yourself through the summary, is very important. Of course, DNA replication is very similar, not during meiosis 2. Number of divisions, very different, 2. Synapsis definitely occurs in prophase 1, crossing over happening. Number of daughter cells in genetic composition, 2, identical clones. 4, genetically different. And then role in animals, fungi, and plants. Yeah, mitosis is used for growth and repair. Sometimes asexual reproduction, depending upon the species. We don't need to know the word gametophyte. That's just like spores and stuff. Don't need to know this either. But reduces number of chromosomes. Sets by half, right? Going to haploid. And introduces genetic variability and differences, which is crazy important for sexually reproducing organisms, okay? And this is just kind of explaining what I just said. So I won't scroll. So if you want to pause and read this over, but it's similar to what I just said. And here's this slide. And here's this slide. Just so much information. Similar information said over and over and over again. So there you go. And then we'll pause right here. And I'll make another video for topic 2. So again, thanks for watching. I hope you have a blessed rest of your day.